We're ready, right? So we're going to talk about the Human Genome Project, and this is actually one of my favorite lectures. So um, this is back in February. So what is this? Is still March. So last month, there's a special issue of Science Magazine about the 20th anniversary of finishing the Human Genome Project. And I will tell you a little bit more about the history of it in a few minutes in the lecture, but basically it started in 1985 and took 15 years to finish. And so it finished roughly 2001. And it's kind of fun because back in 1984, 85, around that time, I was a PhD student. And my thesis advisor, he was a physicist, he's real smart. Physicists are generally pretty smart. If I was a physicist, I would probably be pretty arrogant because, they're very, very smart. But anyway, he went to this meeting in Los Alamos. And so I was kind of tagging along. And in this meeting, these are all nuclear physicists mainly, and other talking with some that are interested in physics and my thesis advisor was interested in DNA structures and kind of the physics, looking at it from a more using physical methods to measure things. Anyway, so they were talking about sequencing the human genome, and remember, I think I showed you Fred Sanger had sequenced this virus in 1977. He got the Nobel Prize for it in 1983. Well, this is 1984, so it's a year later. And they're saying, we want to sequence the human genome. Well, again, physicists are real smart, and they kind of did the math here, and this virus is 5,000 nucleotides long, and it took them 2 years. The human genome is 6 billion nucleotides long, so they're like, okay, it's going to take 1 million years, more than 1 million years at the current rate. And so what they realized is the problem was the sequencing technology. They needed to invest in improving technology. And this is in New Mexico, in Los Alamos, and they basically, the military agreed to spend, Department of Energy, agreed to spend $200 million a year, every year for 20 years. So this would be $4 billion they're going to spend on sequencing the human genome. It was more money than the Apollo space program. It was just a huge investment. And that's what's part of what was behind this big rapid change in sequencing technology, so that now we can sequence. The first genome took 15 years and cost $3.5 million. Now a high school student can do it in 5 minutes for $20. So it's really, really kind of changed things a lot. So I first gave this lecture, like I said, I went to this meeting, it was 84. And so we had a group talk for the research group I was in, and I kind of talked about the meeting and stuff and what it meant. And we had discussions. And then the next year in 85, I was a graduate assistant and gave a lecture and a course about it. And then in 86, I talked about it. And basically, I wound up at least once a year giving this talk. And then more recently, when I do workshops and things, and I've given lots and lots of these workshops, and I always give this talk in the workshop. So the point is that I've probably given this talk 100 times or so, and update it with time, but it's kind of fun. So, okay, so this is looking out our breakfast window. So these are the woodpecker and a flicker, which is kind of in the woodpecker family, as you can kind of tell on the little suet feeder in front of our window. Okay, so here we are, Human Genome Project, and questions. So Andrew works with trees. And so the same is true as trees and humans. If you were to take a piece of a tree instead of humans, but it's the same thing, and you're to take the DNA and shred it up, and you're just to put it in a bucket, and you pick out a piece, what can you say about it? What does it contain? Do you know, Andrew?
It should just contain the genomic DNA, hopefully.
So you take the DNA from a tree. So before you take a sample, extract all the DNA. So this is all plant DNA now. Chop it up and you put it in a bucket, if you can imagine, and just pull out something. What are you almost guaranteed you're going to contain? You would have if you were to pull out a random piece of DNA from trees or humans. Any idea?
No. Probably an intron or an exon.
So let me, you're getting along right. Do you think it would contain a gene? a gene encoding a protein?
Maybe.
Maybe. Do you have any idea what the chances, what the odds are?
No.
If you did it 100 times, how many times do you think you'd get it? If you did it
Maybe
100 times,
50.
maybe one.
Ah.
Less than 1% of the tree DNA encodes proteins. So most of what does it encode if it doesn't encode proteins? Most of it's spacer DNA, most of it's repeats. So for example, in humans, the Y chromosome is you have this pentamer, CCGAT, and it's repeated 5 million times. That's the Y chromosome, CCGAT, 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 really, really boring. And really with the short read sequencing, really difficult. In fact, you couldn't sequence it. That's why the human genome is still not quite finished. It's almost there, but because you have all these repeats, the repetitive DNA is a real problem. So third generation sequencing. Anybody know what that is? I hinted at that in the last talk.
Like the Illumina.
That's second gen. That's called next gen or second generation sequencing. That was brand new in the late 80s, early 90s. That was cutting edge, 1995. But
It's third generation.
Third generation is something now that really, it started around 2015, and they're still kind of tweaking it, but now it's really starting to take off. So this is much more recent. So what we call next gen or Lumina, this is like 25, 30-year-old technology. So now I'm talking something, it's 4 or 5 years old, so it's kind of old, but not that old. So third generation sequencing is single molecule sequencing. And there's a couple of different approaches to that. One is where you actually image the molecules. It's called Pacific Biosciences, and that's a fantastic, it's kind of expensive. And the other is with the Oxford nanopore, where you have these little tiny pores and the sequence goes through there and you can measure a change in the current. The advantage for the Oxford nanopore is it's cheap and it's very fast. Initially, there was problems with the high error rates, but they've got that. It's getting much, much better now. So, and like I said, it's just insanely expensive. It's one of these things that's called disruptive technology. So, for example, we're wanting to sequence COVID-19, and in this project I'm on, we can sequence SARS-CoV-19 genomes, and it takes us a couple hours, and it costs $15 on Oxford Nanopore. This grant that I'm working on with one of these cores here at UAMS, they're all excited. They talked to Illumina, and they got the cost down, and it's only going to cost $70 a sample and take 2 or 3 weeks. And I'm like, well, yeah, okay, that's nice. That's nice for that technology. But here we can do it $15 in 2 or 3 hours. Well, yeah, but that's okay. So we're going to do both. But yeah. So you hear people talk a lot about Illumina and next gen, and that, think 1990s, 95, early 2000s. It was great back then. That was cutting edge 20 years ago, 25 years ago. So next gen is single molecule sequencing, and not only is it single molecule, but you can get really long reads. And by long reads, I mean with one read, you can get something that's 1 million nucleotides long. On average, we get about 20,000 nucleotide reads. And on average, you can get things much longer if you're careful in preparing the DNA. So for example, the SARS-CoV genome is 30,000 nucleotides. You could get the whole genome in one read. And you get multiple reads, and you can make sure you kind of clean it up, and you get errors and other things, but you have to work it out. Now, the disadvantage is because it's single molecule, now you start having to worry about quantum mechanical effects. And so there's a lot of weird things because you're sequencing one molecule. And so sometimes there might be a T there rather than an A, just by chance, but you're only sequencing one. So you need to sequence quite a few to make sure that the error, to get an accurate view of what is what's called the consensus sequence. So that's third generation sequencing. Now you're going to take 2 people, and how different are they in terms of gene content? Would they contain exactly the same number of genes, do you think? Anybody want to guess?
I think it's only like a 0.2% difference.
0.2% of what? Now I'm talking about genes.
Geez.
And it turns out that on average, and it varies a bit. There's a lot of noise, but on average, most people, well, a typical person has about 10 genes completely missing. And they have another 100 genes that don't work. And they have a few extra genes as well that other people don't have. And so it turns out that there's a fair amount of difference. And in fact, there's a larger difference between two humans than there are between two species of birds. And so if you think about in the human genome, they say that the error rate's 1 in a million. I think that's maybe too high, but let's pretend that that's right, 1 in a million. If you have 6 billion nucleotides, that's 6,000 mutations every time your cells divide. And you have as many cells in your body as there are stars in the galaxy, right? So enormous number of cells. So you have enormous number of mutations and enormous number of mistakes. And of course, sometimes you get cancer and other things, right? But you also have differences. And that's why sometimes you can have like mosaic where people have like different, like they have a combination of lighter skin and darker skin. You've probably seen this, and you have all kinds of things. You know, you've seen probably dogs like with 2 different color eyes. That has to do not necessarily with genetic, in terms of genome, but there's sometimes molecules when you're developing, you have these proteins, and they randomly drift. And early on in development, if you have more on one side than the other, this could affect the color of the eye, and so you have different color eyes. So you have a lot of differences between people. And what does 10x coverage mean? If you were looking for something called single nucleotide polymorphisms, you need to look for fairly good coverage, at least 10x coverage, more than that. But any idea what 10x coverage would mean? The human genome is 6 billion nucleotides. So what would 10x coverage mean for 6 billion?
60 billion?
60 billion, yeah. So you would basically need enough to cover it 10 times, right?
Right.
So if your genome is 1 billion and you sequence 10 billion nucleotides, now you covered it on average roughly 10 times. So that's called 10-fold coverage. That's all that is. Okay, so we'll start off fairly simple. What is meant by genome? And originally genome meant all the genes in an organism. And you know, again, the Oxford English Dictionary, what I like is they have, the word was first used in 1930s, and it actually was coined by a Dane. It doesn't matter, but anyway. And again, gene and genus and genome, it's all kind of the same root words, you know, from the Greek beginning. So 1970, Scientific American says that the human genome consists of perhaps as many as 10 million genes. Whoops, sorry. So how many genes are in the human genome? Do you think there's still, is this right? 10 million genes? That's a big number. Does anybody know how many genes there are in the human genome?
Is it like around 23,000?
20,000 is kind of the current estimate, right? And so, you know, originally it was 10 million genes, but then they said, okay, maybe it's only 1 million genes. And for a long time, when they're sequencing the human genome, they thought it was 100,000 genes. And as they got towards the end of sequencing the human genome, they had a pool that you could bet. And the person that got the closest to the number would win the pool. And originally it was like $5 a bet. But as they got closer and closer, then it got more expensive. I think in the end it was $100, $200 a bet. And they said there's only 20,000. Everyone was really kind of surprised. And to be honest, part of it is what you mean by a gene, because you have alternative splicing. You have, it's not as easy as people think, and it's not finished. So one of the things is you need to actually have from telomere to telomere, all G's, A's, T's, and C's, finish the whole thing. Then you have a gene finder that works, that you know is really good, that consistently finds all the genes. Then you can say how many genes there are. Can't really do it until you're finished. And so anyway, that's another story, but we'll talk about that. So like I said, so this came out in February 2021. So this was last month, about 6 weeks ago. Human Genome at 20 says about, and the interesting thing is some of the articles in there, they're talking about modulating gut microbiota to treat cancer. And then hear about super spreading genomes. Here's about viruses and how that viruses can you can look at epidemiological events, and like I said before, viruses can cause cancer in humans. Okay, so this is a little bit of a history about sequencing. So now we're going to talk about first, second, third generation sequencing. So first generation sequencing, and this is from when I was a student, and I thought this was a really old method, Maxim Gilbert, and I didn't really realize that that had only been out for a couple of years. But what you would do is you would run these gels, and you would have a mixture. It's either a purine, a gRNA, or a G. And so this first one is a G. Second one, it's a gRNA, but it's not a G, so it must be an A. That's kind of, so you can kind of guess. And it works, but it's a little bit awkward and crude, but you can get a sequence from it. And then what I thought was a newer method, like I said, I later discovered these are both developed around the same time, but this method is so much better because you have one lane for G, one for A, one for T, one for C. How easy can that be? And so you basically, you have these what's called dideoxy. And so these would stop along. So it would read, but you have these fairly low concentrations. So you get different stops at different G's. So it stops here, here, here. And so you could basically then get the sequence from this. And this is done by Fred Sanger. And he got a Nobel Prize for this, but it was actually his second Nobel Prize. He got the first Nobel Prize for sequencing proteins. And I gave a talk in Cambridge in Fred Sanger's lab, and Bart Barrow was there. Bart was a postdoc in Fred's group. And Bart told me that he got his PhD for sequencing 53 bases of DNA. And I was really impressed. I said, 53 bases. He said, yeah, he said, if Fred Sanger can get the Nobel Prize, he got the Nobel Prize for sequencing 5,000 nucleotides, 50's worth of PhD. So I thought, okay, that's probably true. So, and this is a nice picture. Fred also has, you see other pictures of him with a little bit more of a bushy face. And Maxim Gilbert, Fred Maxim and Wally Gilbert at Harvard. So the two Cambridges are Cambridge, Massachusetts, and Cambridge, England. This is where these were developed. Okay, so this is the. So let me just back up here. This is looking at. This is again, we were talking before about Maxim Gilbert, and versus this is the Sanger method where you get one for each sequence, and here you get a little more complicated. There are reasons you probably maybe want to use. So Maxim Gilbert sometimes can resolve some structures and get compressed and things, but it's just it's not so good. So here's the gel that I did, and this is 1984, something like that, 1980s. As part of my thesis project, and I had cloned this insert into a plasmid where it went GA, 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 TA, GA, 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 GA. And this could form triple-stranded DNA. And so we were looking at probes and things. And so this is what sequencing looked like in the 1980s. And there's actually a machine that can do this. This is taken back in 2012, but this is basically, you can do what's called Sanger method. And so the Human Genome Project, like I mentioned before, it started in 1985. And it was, the US government agreed to invest $200 million a year for 20 years. The human genome is 3.4 billion bases for haploid, but remember we're diplogens. You get one from your copy from your mom, one from your dad. So it's 6.8 billion. So if you were to read one base every second, G, A, C, G, A, T, it would take 216 years to read the sequence from one person. And that's from one cell. And remember, every time a cell divides, you're going to have 6,000 mutations or so. 216 years. And all you would know, there's a couple of things. First of all, you'd be really bored. Like I said, if it's a male, CCGAT repeated 5 million times for the Y chromosome. It's really, really boring. You'd be very bored. And also, you would know that this person would be dead because most people don't live to be 216 years old. And so I said that to say you're going to have to use computers to be able to handle this. So back in 1970, it wasn't even possible to sequence the human genome. By 1980, they had mapped 3 genes. It was going to take 4 million years to sequence the human genome. By 1990, they mapped 12 and maybe 1,000 years. By 2000, they had a draft. In 2001, they said they were finished, but it wasn't really. It was kind of a draft. 2010, Now they say there's 43,000. You could do it in a few hours, but the number of genes is still kind of not really sure. Like I said, you have to finish it first, then you have to have a good gene finder. You can be consistent, look at expression and figure out what you mean when you say gene. So, and so, like I said, I've been giving this talk. So 2011. So then in 2010, I did the talk. Then I added, okay, 2011, one human genome was being sequenced every 3 minutes. It was 175,000 genomes a year, 2011. That was 10 years ago. There's this 1,000 Genomes Project, which at the time was a really big deal. By 2015, one genome is being sequenced every second. But the problem is there's no really common storage. We don't know where exactly how many there are because there's no place to store it. So now, 2019, getting a little bit closer, the first complete human chromosome was sequenced. And like I said, so they're getting almost finished now. By 2021, they're almost there with a complete set of all 23 chromosomes. It'll probably release soon. And they've actually, this first chromosome, they sequenced a really small one, and now they're starting to sequence bigger ones. And most of the chromosomes are just in a few pieces, like 3 or 4 or 5 pieces. So it's probably by this summer, they'll be finished. So the first human genome cost $3 billion, took 15 years. The second genome, this is also Craig Venter sequenced himself, and it only cost $100 million and took 9 months. They sequenced a few years later Jim Watson's genome for less than $1 million. And it took 2 months. And now, so this would be like John Doe, and in Denmark, Jens Jensen is a very common name. So you could sequence a person for $1,000 and it would take a couple hours. And now, like I said, today in 2021, you could sequence a human genome in literally a couple minutes for $20, and high school students can do it. So it's really just amazing. 2009, they said that they could sequence a human genome in an hour. Again, this is a company making this claim, and it didn't really work. PacBio said that they could do the human genome in 4 minutes in 2010. They can almost, do it now in 10 years later. It's just, there's been a lot of problems with this. But Oxford Nanopore says they can do it in 15 minutes in disposable stick. They can now. They couldn't back in 2012. This is a sales rep, but they're getting there. I think that we really are getting this technology. So this is what's called a next-gen, second-generation sequencing. And one of the early ones was a Roche. There's a 454 is the name of the machine. That's just kind of the company originally that had this. And it's using basically automating the sequencing. And this is basically describing how this works. And this is what's called next gen or Lumina, or this is what everybody uses anymore. Any sequencing by default, this is what they're going to do. And it's really revolutionized things because it is so cheap and it's really fast. And basically, you have a microbead with a piece of DNA, and what you do is you're going to do an emulsion PCR and amplify this up, and then you got your little wells in here, and what you're doing is you're basically you have a primer and you're sequencing along. You chop the DNA up, and you're going to get. You have different colors for different nucleotides, GATC, and you take pictures of each of the cells, and you can see there's a G here or an A or whatever, and you take lots of pictures over time, and you can from that get the sequence. And the images that it's like gigabytes of images, and yeah, well, anyway, that's pretty technical. But the bottom line is that so Moore's law. Basically, I don't know if you guys know Moore's law in electronics, but back in the 1960s, they discovered that as they put more transistors on a chip, it's more efficient, and that roughly every 18 months they could double the number of transistors you could put on chip, and that still holds true even today. And so what happens is roughly every 18 months you can double your processing power. And so Moore's law is this line here. And for a while, the sequencing cost, the drop was following Moore's law because you can kind of automate things. And then there's this sudden plummet here. I'll come back to this, what's going on. But I just want to explain this y-axis. So this is how much it would cost to sequence 1 million bases of DNA. And that's roughly bacterial genome. So back 20 years ago, it cost $10,000. And then by 2005, it cost $1,000. And then by 2007, see, it was still kind of going along the lines of Moore's Law, but then it started dropping here, 2008, 2009. By 2010, now it costs less than $1 to sequence a genome. And by 2015, it cost a few pennies. So what's happened here is, and there's more recent versions of this, and it does continue to go down some, and we'll come back to this, but what's happened here is this is when the Illumina second generation sequencing really started taking off. They started mass producing these, and the cost really dropped a lot. And so now you could sequence quite cheap. Now, third generation sequencing, there's a variety of different ways of looking at this. There's something called basically, you can actually take pictures of this, and this is what's called PacBio, and zero mode wavelength guide. And basically, what you do is you've got your double-stranded DNA here, and you're synthesizing, but you can actually, as you go along, you can look at the nucleotides that are being done, and so you can do that very with each nucleotide. You can kind of follow it. Now you can also, with the PacBio, they've got a very thin thread. They could actually drag this along the DNA with a very thin needle, essentially, and image it, and you actually get pictures of that. I'll show you that in a minute. The Oxford nanopore, what happens is you basically have a piece of DNA and it's flowing through a pore, and you have a current here, and as it goes through, it changes the current, and you can from that figure out what it is. So this is the PacBio machine. This is an older version, but they're really big, and they're usually in the basement because they're very sensitive to vibrations, because what happens is you're actually measuring molecules. You're taking pictures of molecules. And so it's fantastic. It's single molecule resolution. I don't know recently, but I know that a couple of years ago, one run would cost $20,000. So it's kind of expensive. And you need to have this big liquid nitrogen, and it was just this really big, and the machine itself cost $800,000 for the machine. And then every time you run it, it's 20,000. And so that's one way. That this is called third generation. Now the advantage for this is you could get reads that were 5,000 nucleotides long. And Illumina reads are only 100, maybe 200 nucleotides long. And so you get much longer reads. And they had this way where you could they could put it in a circle and you could read through and get really accurate reads that are really long. And so that was a big advantage for that. And then this mid-ion came along. This is back in 2012 when it first came out, the press release. They say they could do the human genome in 15 minutes on a thumb drive. And here it is. You basically plug it in, and that's your sequencing machine. And that was the idea. And it's a fantastic idea. And they said, okay, it costs, $900. And they can read 10,000 nucleotides at once. And 10,000 nucleotides is fantastic, particularly back in 2012. Now, like I said, routinely, we can get 20,000. You have to be careful how you prepare it. If you prepare your DNA very carefully, you can get much longer. You can get 500,000 nucleotides. So Oxford nanopore, this is called third generation sequencing. And the first generation, Sanger, to sequence a bacterial genome would take about 2 months, 60 days. With the next gen sequencing, Illumina or 454 would take 2 days. With Oxford nanopore, so this is historically, and so now Illumina is still, early on was about 2 days, and even on a high seq, it still take a couple of days. If you did a MiSeq and you did things right, you could probably get 1 day, maybe even less than 1 day. But Oxford nanopore, you can do it in a couple hours, do a bacterial genome, and we've done that. And usually, in fact, for this class last time, 2 years ago when we did it, the students and 2018 and 2019, the students sequenced genomes. And what happened, I would give my first lecture on Monday, where the students would actually load some samples on. I would give my first talk. We would break for lunch. After lunch, we would give students some of the reads, and they could actually then put together and figure out what their bacterial genome was. So it doesn't take that much time. And it's very portable as well. This is your own machine. And it's fairly cheap. So the Illumina is going to cost around $100 or so. Originally, of course, Sanger would cost $10,000. And back in 2005, it's going to cost $1,000, but now that it's, you know, it's maybe around $100 or so. Oxford Nanopore, you can buy this flungal and you can do barcoding on it. The flungal is $85 and you can do 12 samples. So, you know, it's in the range of $10 a sample. So that's not bad. It's also fairly high throughput. So you can get 15 gigabases. And again, this is an older slide. This is from 2012. You can get more than that now. In fact, the machine, I have an instrument grant for an Oxford Nanopore Promethean machine that can generate terabases of data at a time. And in fact, I think over a period of a couple of days, you could generate a petabase, 10 to 15 bases with one machine. So you can really generate a lot of this. And so, of course, Illumina, you get 100 gigabases, but these are also, you need to assemble, and you also need to do library preps beforehand. And then once you're done, you have all these pieces and have to put it together. The nice thing with the Oxford Nanopore is you have really long reads. So, and because it's single molecule, if you do it right, you don't need to cultivate, but you also don't need to worry about assembly because you have these long reads, which is really a big thing. And so this article is talking about, this is by Mark Gersten at Yale, and he's talking about, Mark often gives talks about this, how that although they talk about the $1,000 genome, and isn't this really great, it actually costs a lot more than $1,000 to analyze it. So I've shown you this slide 2 or 3 times already. It keeps kind of popping up, and I apologize for not editing out. This is next gen, and here's kind of third generation sequencing, and you can really see the difference here. Okay, I'm going to already showed you this. Now, it used to be back in 2009. So I gave these lectures for many years, and then what I do is I would update it, and then eventually I realized, okay, 2009. Here's how many genomes there were, and then I would say 2010. Look, in 1 year it's grown so much. So back in 2009, there were 2,000 genomes, and now we're discussing this in class. There's 300 genomes, 300,000 genomes. So literally, that's grown 100-fold in 10 years. In the sequence read archive, you can actually look to see how many human genomes there are, and you wind up with saying that there are 3.5 million 17th February. So this is from an old slide back in 2005. And I'm not quite sure why they chose 2005, but back in 2005, I guess it was a couple of years after the Human Genome Project was finished in 2001. And by 2005, they come up with these new machines, and they could basically sequence, if you had 10 of these machines, you could sequence the human genome in a couple of days. So one machine, you could sequence 0.6 gigabases, or a tenth of the human genome. It would take 40 hours. By 2017, you could sequence 6,000 gigabases, or basically 1,000 human genomes. Right? So you've gone from a tenth to 1,000. So it's a 10,000-fold increase. It's 2017. The Human Genome Project, depending again on who you talk to, it's 13 years, $2.7 billion, or 15 years, or 3, whatever. It involved 200 research groups, just an enormous amount of effort, a lot of time. And to sequence the genome now costs, you can do it in a little more than a day, or this is in 2017, $1,000 one operator. And this is where most people are still at. And if you go to UAMS, they have a sequencing core, and this is what they will say. Now, this was 2017, so this was 4 years ago. If we update it, we say, okay, now in 2021, with these Oxford Nanopore, they have a new version of these flow cells, the earlier ones that we're looking at, and the ones that we would use for this class. These are the flungal ones that cost $85. They have 500 of these little flow cells, 520 flow cells. Actually, I used to have one I'd hold up. When I give lectures, I'd always hold it up along with my Swiss chocolate bar and everything else. So one of these sequencing machines, like I said, would cost $85, and it has about 520 flow cells. Now, the next version is a little bit more expensive, it's about $1,000, roughly. But it has on it, instead of 520 flow cells, it has 1 million flow cells. and that they're estimating the price of this is going to drop. But anyway, the bottom line is you have 1 million flow cells, and you can see what's 1,000 bases per second. So that means you could do for each flow cell. So you can sequence 1 billion bases in a second. So you could sequence for one of these machines 144,000 gigabases, which is a millionfold increase. Instead of 24 hours, if you could do a base every second, you could do 1x coverage in 3 seconds, or cover the 1x haploid, or for really 6 seconds to do the whole genome. But basically, within a minute, you'd have 10-fold coverage. And $1,000, it would cost $20, and you need a high school student, and you could do it. I mean, it's so it's just really amazing what's happened. This is what's called disruptive changes. So using Illumina, using next gen, second generation sequencing, they had estimated that by the year 2025, we would have 40 exabytes. And again, this is 2015. So now we're halfway there, a little bit more than that, or 2021. And we already have more than 2 exabytes just in last year alone. To store 40 exabytes of data is going to cost $4 billion. And, you know, it looks like by 2025, we'll have 20 zettabytes, 10 to 21 amount of data. So that's an enormous amount of data. So it's enough to keep us busy a while. So this is a RIN. Okay, so I'm going to talk a little bit about now a different, we're still talking about 3 generations, but now I'm going to look at length. So a bacterial gene, on average, is about 1,000 bases. And first generation sequencing, it was around 600 or so, 600, 700. And that's the gel that I showed you. And we could run really long gels, and we could get, you know, maybe 600 bases or a little bit longer, depending on how we do it. But typically, on average, we could get at least 600. Now, and so that's 60% the length of a gene. Now, when Illumina first came out, the reads were 35 nucleotides, not 600, so a lot smaller. And they got a little bit better. They got 50, and then they got 100. And now they're 100, 120. You can get 150 base pair reads and maybe 200 base pair reads. Those are long reads. So second generation sequences are currently still about 200 bases. And so that's 20% the length of a gene. This sometimes causes problems because they're so short. You have to stitch them together, and you can do it. You can assemble it, assuming that you don't have too many repeats. For bacteria, you can get by with it. That's why they sequence so many bacterial genomes. For the human genome, this is really causing problems. That's why it's not finished yet. Third generation sequencing, the reads, like I said, on average are about 20,000 bases. So you can fit easily 20 bacterial genes in there. And so here's a gene. A gene is only 1,000, and you can read 20,000. So you get the whole gene, and not only you get the whole gene, but you get all the genes next to it. So this is really, really powerful. This is really, really important, I think. And so oftentimes in bacteria, we use, remember I talked about using ribosomal RNAs, and the 16S ribosomal RNA, or with the whole operon, including, is around 5,000 bases long. 16S is around 1,500. So with third generation, we get the whole thing easily. With second generation, you only get 100 bases or 200 bases, and so you don't get the full length. And what happens is, because the ribosomal RNA operons are repeated, you can never completely assemble genomes. And so the reason, you remember, you look, 90% of the genomes are draft. They're not finished. And the reason that they're draft is because of this inherent problems in second generation sequencing. The reads are so short. When you come up with these repeats, you don't know whether this 200 base pair went to this one or another one somewhere else in the genome. And so we actually calculated, and here's some things looking at the different genomes and looking at coverage and things. And we actually did some simulations, and basically, even if you had 1 million-fold coverage, you still could not assemble these bacterial genomes if you have lots of ribosomal RNA operons. You just can't do it because the reads are too short. These reads are so short. And early on, this must have been 2001, 2002, so about 20 years ago. A friend of mine, collaborator, gave me some of these next-gen sequencing from he'd sequenced, I think, a couple hundred bacterial genomes. This was, I don't know now, it was 2002, is right after, remember the anthrax letters after 2001, the bioterrorist attacks, someone had sent some letters with anthrax. And so this guy, the military was very interested in this. And so they bought him one of these machines to sequence one of these next generation, second generation sequencing. And he gave me a bunch of bacterial genomes. And I took it back to my lab. And I was real happy because at that time, this is 20 years ago, I had like 100 bacterial genomes, which is more than was in GenBank. And my students started complaining. And pretty soon there's mutiny in the lab. And I was like, oh, shoot. And so I sat down and tried to troubleshoot. And I figured I would listen politely to the students and explain to them why they were wrong and then solve the problem. And I listened to the students, and the students were saying, look, these reads are short. I'm like, yeah, I know. But you should be able to assemble them. They said, but the problem is that there's high error rates at the end and you get a lot of truncated genes. And I'm like, yeah. And then they're explaining the problems of truncated genes. And eventually I listened to all the problems and I realized this is not going to work. There's just some inherent problems in this. These reads are simply too short. Here you have something, you're only reading 20% of the gene, 20% of the length of a gene. No, it's not going to work. And so eventually I told the students they're right. And we only worked with complete genomes. And I got a lot of flack for that. And in fact, I lost the promotion because I didn't do next gen sequencing. But, I want to be able to do things. I'm interested in complete genomes. And it's a real pain in the butt to actually finish genomes. It's very expensive. Again, that's why only 10% of them are done. It takes a lot of time and effort to be able to really clean it up. Third generation sequencing, keep your fingers crossed. Hopefully you can get a nice complete genome. And I think we're getting close within a couple of years of being there. Okay, so that's a little bit about length now, which brings us to the next part about why is a human genome not finished? And you say, well, this is a 20-year, it was finished 20 years ago. No, they had a draft. They declared it done, but it was in lots of pieces. And it was in tens of thousands of pieces. So it was nowhere near done. And if you look even here, see these scaffolds, 15,000 pieces, 5,000 pieces. And this is considered good, high quality. This is like the best they've ever had. And it's only 15,000 pieces. This was done last October. And so here, this one, this is kind of fun. It's 24 scaffolds. And if you look, it's nearly there. These scaffolds are close to being done. In fact, I think a couple of them, 2 or 3, are actually 100%. And there's this project called telomere to telomere, T2T, where they're actually they're working on trying to finish. And they're using a combination of PacBio and Oxford Nanopore to sequences. And so this is as of February this year. And so here's looking at scaffolds. Okay. So, yeah. Why is Human Genome? Oh, it's just now finished. It's almost finished. So this is in January of this year. So really, and so this is an article looking at, and again, this came out in February, last month. Genome assembly projects get a boost for high accuracy long read sequencing. And so see, there was some problems because the genomes weren't finished. And so they're trying to finish the gaps. And this is called telomere to telomere, T2T consortium. And they're actually trying to and I know this guy, Adam Philippe, and basically a lot of people have been kind of complaining for years, it's not finished. We need to actually finish the genome. And so this is again about the telomere to telomere consortium. Yeah, so that basically they're getting there. It's all, and this is really kind of fun. They have a bioarchive version, and basically they've got one contig for each chromosome. And like I said, they're using a combination of PacBio, Oxford Nanopore, and Illumina. And they're doing all three kind of together and getting pretty good pieces. So this is really good. Now, but the question is, getting back, why is the human genome? Okay, so it's almost finished now, but the reason it's not finished partly is because it repeats, but also the question is, why is the human genome so large? And so here, if you have a bacteriophage, this is bacteriophage lambda, and this is a virus that infects bacteria. And if you were to type it out, it'd be about 2 pages of text, G's, A's, T's, and C's. If you were to look at the E. coli genome, again, just as letters, it would be a book that's about 200 pages. And then here's yeast, and it's a little bit thicker. Again, this is a eukaryote, a very simple eukaryote, but this makes bread and wine and other things, right? This is yeast. It's around 12 million bases, has 6,000 genes, about the same number of genes as coli, But again, this is a eukaryote, and so it's 50% coding. It's about 500 pages long. Now, if you go to an animal, you look at worms, or Andrew, here's Arabidopsis. You recognize that, Andrew? What is this?
Arabidopsis Thaliana. Yes.
Ah,
My plant.
that's a plant. Yeah, absolutely. So the botanists love this plant, right? Because it's ideal. It grows in the lab really easy. So if you were to take the genome sequence for Arabidopsis, and it's also relatively small for a plant, it's about 100 million bases, and it would take 3 volumes, 3 kind of thickish volumes. Okay. So now, if you were to compare this with the human genome behind me, you've seen some of these pictures where I'm on my camera. I have the Encyclopedia Britannica, right? So the human genome is this huge set of encyclopedias. It's 80 volumes. It's huge. And biologists say, well, of course, because humans are so complicated. And so this goes back again to Aristotle. We started off talking about Aristotle and life. And so Aristotle's ladder here of complexity. So at the top of the ladder, you have humans. Does anybody recognize this guy? He's a human, but any volunteers for who this guy is? He wrote a lot of fairy tales. Like The Emperor's New Clothes and other things. This is Hans Christian Andersen. So he's a famous Dane. That's why we don't have to do my proof. And so you have humans at the top, and they're the most important. And of course, close to humans, you have cats. The Danish word for cat is K-A-T. Just so you know, anyway. So I said, oh, that's just like in Arkansas. That's how we spell cat in Arkansas. So it was pretty easy for me to learn. So you have humans and cats at the top. And then below humans, you have mammals. And then below mammals, you have birds. And below that, you have reptiles. And then you have fish. And you have insects. And then you have things like jellyfish. And then you have higher plants and lower plants. And then, Aristotle, you had crystals, you had minerals as well, but we won't talk about minerals. But so you have this idea. Humans are the most important thing and the most complicated. And as you go further down this way, things get more simple. And so as I started sequencing genomes, we started collecting, and even before they were sequencing genomes, you could estimate the size. This goes back in the 1960s, 1970s. You could figure out how much DNA haploid an organism has. You could do some experiments by looking at hybridization. And so, but as it began to sequence genomes as well, you could kind of combine this data. And so what we did is we had a bunch of different organisms here, and I divided them into archaea, bacteria, fungi, protozoa, plants, and animals. And so then we can ask a very simple question: how big is a genome? And so we have all these different collections, and you get a range here. So the little bars represent kind of from the smallest to the largest. And there's a nanoarchaea that is less than 1 million. It's only like 500,000 nucleotides long. And do you remember we're looking at the smallest bacteria is only 100,000. So the smallest bacteria here is 100,000. And some archaea can be up to 5, 6 million, maybe 10 million. Some bacteria can be 12, 15. Remember, 16 million was the biggest when we looked at the record last time. So they can range from 100,000 to 16 million. So that's a pretty big range here in size. This is a log scale. Fungi, so some fungi can be small, quite, they can be maybe 10 million or so. Pombe here is 12 million. Yeah, 12 million nucleotides long. It's because of Saccharomyces pombe. And it's also Saccharomyces cerevisiae is also around 12 million or so. And some of them can be, so this is 10 million, 100 million, a billion. And so it's less than a billion, but you can have several 100 million nucleotides for yeast. Then protozoa are interesting because you've got some protozoa that are only a couple 100,000, but you have some amoebas. In fact, some amoebas are 10 to the 12th are trillion nucleotides long. That's 1,000 times bigger than the human genome. Kind of interesting. And then you've got in plants, you have moss, which has only got 10, 20 million, something like that. Arabidopsis is here. Andrew, what was the size of Arabidopsis? Do you remember? Was it?
think it's 2.62
Was
megabase
it?
pic.
No, it's got to be bigger than that. Probably 20 or 30 million bases.
It's 100 megabases.
100
Oh
megabases.
no, it is 30.
30 megabases. Yeah.
No.
And so some plants are really ferns, it turns out, are, so this is a billion, this is 10 billion, this is 100 billion. Some ferns are 100 billion, more than 100 billion nucleotides. So the plant size really ranges quite a bit. And now we get to animals. So ticks are basically, some ticks are about the size of some large bacterial genomes, in the order of, I think, 18 million bases for ticks. Fruit flies range from about 80 million to 800 million, roughly, in that range. And this is an interesting story. I'll come back to that later, but they vary in size depending on where they are. Humans are roughly 6 billion, right? Cockroaches are 9 billion. And lungfishes are 100 billion, more than 100 billion. And this is kind of interesting. So fruit flies, it turns out that the size of the fruit fly genome depends on where they live. If they live in the Andes Mountains up very high, they have a small genome. If they live in the ocean, they have a larger genome. I'll come back to why that is, but that's kind of interesting, looking at size. So my point is, humans are not, they don't have more DNA than anything else. In terms of plants and animals, they're kind of more or less than the average. So if we go back to this database of genome size, but now what we do is we're looking at, again, this is Aristotle's ladder of complexity. So the amoeba wins 1 trillion nucleotides for haploid genome. Ferns comes next, and then you have lungfish, then you have cockroaches, then you have humans, and then you have fruit flies. So humans are somewhere between cockroaches and fruit flies in how much DNA they have, which is a little bit, I gave this talk at a small liberal arts college, and some of the religion professors were kind of upset with me for, they said, why would God do this? I said, oh, I can't really speak for God. It's kind of like on that Dragnet show, just the facts here. Sorry, this is just the numbers. Don't shoot the messenger. But humans, basically, the human genome isn't really that big by comparison. It's kind of what you'd expect, kind of medium. Lungfish have a lot more. What cockroaches? They have 3 times as much DNA as humans. It's like, really? Why do they need so much DNA? And ferns, just an enormous amount of DNA. And so this difference between how much DNA you need for haploid genome and the complexity of an organism, this was known from the 1960s. It's called the C-value paradox. And the C-value comes from constant or characteristic size of the genome for haploid genome for a given species. So the C-value is basically the size of the genome in terms of bases of DNA is the haploid DNA genomic set. That is how much DNA is in an egg or a sperm. Okay, fairly easy to do. What you could do is you could extract for a lot of organisms egg or sperms, and you can do some things to say how much DNA is there in one sperm, one sperm cell, one egg cell. These large difference in genome size without any seeming relation to an organism's complexity is called the C-value paradox. And for, like I said, it's been around, it's known since the 1960s. And there's a couple of possible explanations. The most reasonable explanation is the concentration of DNA is constant. And so if you have more oxygen in the environment, you can have bigger cells. And if you have bigger cells, you need more DNA if the concentration is constant. So in the fruit fly example, when the amount of DNA that a fruit fly has varies from 100 million to 800 million, the fruit flies that live near the ocean, there's more oxygen and their cells can be bigger because more oxygen can diffuse through there. And if you actually look, you can measure that. And they have 800 million bases of DNA per haploid genome. Up in the mountains, there's less oxygen, the cells are smaller, and now you have less DNA. So in that sense, the DNA is part of the function of DNA is partly a packing material. It's so concentrated, it's around 10 micrograms per mil, and that range is kind of like this little, I don't know if you ever do agarose gels or not, but it's like like a 0.8% agarose gel. It's kind of this little bit of a snotty mess. It's not quite as solid, but it's pretty gooey and gelatinous type. So the DNA is actually an important structural role in the cell, not just making proteins. So just so you know, so here's a fern. This is a fern in our backyard. So what does all this DNA do? In bacteria, a typical bacterial genome, 96% or more codes for proteins, and then 4% is the intergenic region that's involved in regulation and other things. And so, like I said, bacteria, because they can divide really quick, they've had lots of time to really optimize their genome and to really streamline things. And what bacteria do is they have genes that do similar things together. So for example, all the genes that are involved in breaking down a sugar, they're all together so that they get expressed and all regulated. And it's really organized and it's manually coding and it's really, really optimized. In contrast, humans have around 1,000 times more DNA, but they don't really have that many more genes. Now, I don't know if we really have 23,000 or not, but if you think about it, some of these bacterial genomes have 20,000 genomes, or 20,000 genes, or 16,000, 15,000 genes, proteins from one bacterial genome. So 23,000 proteins is not that different than a bacteria. It's like, that's a little bit interesting. Only 1% codes for proteins, and the rest, a lot of it, 45%, is repeats, simple repeats. And then you have some highly repetitive DNA, which we'll talk about a little bit later, and then you have some other types, alu fragments. This is a 300 base pair. It's one of these transposable elements that moves around. You have like 1 million copies of this. And then you have other types of repeats, another 43%. So simple repeats would be like CAG, CAG, CAG. The Y chromosome, CG, GAT, whatever, repeated 5 million times. It's just really simple repeats. So most of the human genome is repetitive DNA. And so you can have different types of repeats. You can have, like I said, it's been known since the 1960s. The connexity association, you've got fold back DNA. So these are some of the repeats that can be things that are palindromes and stuff that can fall back on itself. You have high repetitive DNA, middle repetitive DNA, and single copy DNA. And like I said, this is published in 1968 in Britain Comb. So the repetitive DNA can be either localized in particular regions around, or it can be dispersed, and it depends on what it is. So as an example of this, dispersed, scattered throughout the chromosome, is the alu family. And the alu family is about 300 bases long, and there's 500,000 copies in the human genome. It makes up 5%. And this is dispersed throughout the chromosome. It's all over the place. And these actually move around still. I'll come to that in a minute. These are mobile elements. You also have localized, highly repetitive. And these are things that are, ranging 2 to 10 bases long. There are millions of copies, often in blocks, that make up 6% of the chromosome. They're associated with heterochromatin. Sometimes they're in centromeres. They're very AT rich. And so you have things, for example, that go AT, just repeating, hundreds of times. So there's something called Fragile X syndrome. I don't know if you guys have heard of this or not. But Fragile X is where there's a repeat. And if it amplifies too much, what happens is, and it can, it's the way that sometimes it can kind of amplify when you having replication. And if it amplifies too much, now the chromosome is unstable and it breaks off. That's what's called Fragile X in the X chromosome. And it's actually one of the most common forms of mental retardation. And it happens on the X chromosome, and it often happens in males. So this is looking at mobile elements. And so remember what we're talking about, alu, these are examples of mobile elements in it. And here, here's alu. And there's something else called L1, which is another mobile element. And then you have these SVAs, which are structural variant elements. And so basically, they're looking across, and what they found, this is really amazing for me, they're looking at ALU, L1, SVA, retrotransposition sites. They found out of this, these are for humans, out of every 21 births, okay? What happens is, based on looking at the insertions and the other things, they find that you have genes that can code for proteins, And these transmittal elements can come and change the coding sequence for genes roughly 1 out of 21 births. I mean, that's pretty amazing to think. And so, like I said, humans are very different from each other, more different than two species of birds. And most of the genes in humans are expressed only in the brain. More than half the genes are expressed only in the brain. So we shouldn't be surprised that we think differently. In fact, sometimes we should be surprised that we can think similar enough to actually talk to each other and get along and explain things. People are different. And so, you know, one of the things, this article kind of really bothers me, that they're looking at loss of function. And any individual has about 100 of these loss of function variants. So what is an example of that? Well, for example, color blindness. Well, you know, if you're colorblind, and actually that's an example where it's completely lost. So you're missing, you know, you have red, green, blue receptors and you're missing a receptor. Now, what a lot of people don't realize is, although you have, because you have these 3 receptors, they're very similar, these 3 cone receptors that are sensitive to different types of light. You could have, when you're having chromosomes when they're aligned, they can get off. And so you wind up with a deletion of one of those, right? But you could also get an insertion of one. So some people can see red, green, blue, ultraviolet. And some people can see infrared, red, green, blue. And some people can see 4 different colors. And some people can actually see 5, a few, you know, in some of the artsy fartsy type. And so it's kind of interesting because we have all these loss of function variants. We have, and for me, it's very easy. I've lost the gene that basically says stop talking. I just keep talking forever. And that's a loss of function. That's just something that happens. But this is kind of really interesting, looking across humans and looking at variation. So this is the human genome at 20, and they're talking about some of the what we've learned in the past 20 years. And then one of the things they talk about is data sharing. And this guy, Robert Cook Deegan, is a really famous guy. He's written several books about the history of the Genome Project, and they talk about these Bermuda principles and the need for sharing things. And this is from a science paper from 2019. So January 2019. So this was 2 years ago. And there's a whole bunch of authors. And so you see I'm down here at the bottom. And a whole bunch of these guys were all basically saying what we want to use for the public genomic data, we need to be able to have access. And here again is these FAIR standards: findable, accessible, interoperable. And basically, there's some problems with the loopholes where basically people say, well, share our data as long as you give us credit for it, and then you can't publish it until we publish it first. And sometimes I've been in this case where I had to wait sometimes 2 or 3 years to publish something because the people that sequence it didn't want me to do it beforehand. And it's just, you know, what you should do is you sequence it and it should be in a database that's available, and you can get credit for sequencing it. Here it is, you have credit, but it should be available. Anyway, that's particularly if it's funded for with public money. So that's the end of that. At 10:30, that's not too bad. This is a plum tree from our backyard. You can see a little insect in there. Okay, so now hopefully you can answer this question. If your randomly selected piece of DNA, what does it contain? Anyone want to answer that? Imagine you have a bucket that contains, you take the human genome and you print it out, and you take, you know, print it out on a long strip, just letters GATC, right, a long line, and you kind of rip it up into pieces and you put it in a bucket and you grab it. You look at the bucket and you grab a piece. What can you say about that DNA if you're to randomly piece of DNA?
Probably doesn't code for anything.
It doesn't code for anything. And what else? The sequence you have is probably found somewhere else as well. It's repetitive. The human DNA is highly repetitive. And people don't realize this when they're looking at SNPs, like the SNP database. They think that, oh, out of this 100 base pair, this one base in the middle is changing, so they call it a SNP. But they don't realize that because it's so repetitive, you're going to have this 100 base pair piece a million times. And sometimes that middle base might be different, but it's not necessarily a SNP. It's just that you're looking at different parts in the genome. And people think the human genome is random, and it's not at all random. It is not random. It's really boring. It's the same thing repeated again and again and again and again. And so their assumptions are wrong, and they wind up making sometimes some crazy things. You probably heard, oh, we found the gene that causes homosexuality, and then they say, oh, it's not really, sorry. We found the gene that causes alcoholism. Oh, well, a little bit later. Sorry, no, we didn't. We find the gene that causes breast cancer. Well, kind of, sometimes maybe, but because it turns out it's not so easy. The gene that causes this or that or the other, it's complicated, and there's lots of. So anyway, just so you know, it's not nearly as random as people think. Third generation sequencing technology. Anyone want to guess what that is?
Single molecule sequencing.
I missed the first part of that. What was that?
She said single molecule sequencing.
Single molecule, yes, that's correct. And what is the advantage of second versus third, or third versus second?
The length of the reads.
The length of the reads is really, really important, right? Yes. Okay. In terms of gene content, how different are two individuals?
I think you said like 10%.
Not really 10%, but each person on average basically has around 10 genes that are completely missing and another 100 genes that don't work. And two people, they would have a different set of 10 genes that are missing and a different set of 100 genes that don't work. And that varies some. Some people have 2,000 genes that don't work. In the case of cancer, what happens is you remember where the whole chromosome kind of gets broken up into small pieces and rearranging. And so, yeah. So the point is that there's a lot of variation, even within the same person. So if you were to take the DNA from your nose and the DNA from your kidney, and you're to sequence it, you might see differences, quite a few differences. So the point is that two individuals, they're not at all the same. They're going to have quite a few differences and variability between them. Okay, and 10x coverage means what? If you have a genome that is 10 million bases, what would 10x coverage? How much would you need to sequence for 10x coverage?
100.
100 million, right?